Hi everyone and welcome to today's Biology Daily Booster. This is number 5 in our series for paper 2 and it is an advanced informationistic topic both for foundation biology and higher tier biology, so important for everyone today. What we're going to look at the start of this little booster then is just a bit of a recap on what we said in our previous booster about the numbers of chromosomes in different types of cells. So what we've said is in a normal body cell, then you have 2 sets of each chromosome. Now, in humans, that means there are 46 chromosomes. Beware though, because that's only in humans. They can give you questions on the exam paper about any random species they fancy, so don't fall into the trap of just automatically going for the numbers for humans. Read the question. If it says body cells, it's obviously going to have 2 sets of the chromosome. So if the question tells you how many chromosomes are in a body cell, it's going to be asking you to work out how many are in a sex cell, and you just divide it by 2. If it's the other way around and they've given you how many chromosomes are in a sex cell, and you need to work out how many are in a body cell, times that number by 2. The key words we've got here then, diploid, just mean 2 sets of chromosomes, because remember, if we break that up, di just means 2. Sex cells, the sperm and the ova, have 23 chromosomes if we're talking humans, and they are what we refer to as haploid. So remember, haploid, it's got the same HA as half, so half the number, 1 set of chromosomes. What actually happens in the process of fertilisation then is 2 haploid gametes, a sperm and an egg in the case of humans, are going to join together to form a diploid cell called a zygote. Now, what we're really talking about is the fusion of the 2 nuclei. So fertilisation is where the nucleus of the sperm joins with the nucleus of the egg, and that makes our diploid cell called the zygote. That zygote then undergoes many divisions by mitosis to produce a new organism. Remember, mitosis is going to make 2 genetically identical cells. So what we're looking at there is every division we get 2 genetically identical cells for each one we started with. Now, the other part we need to remember is that every organism that is produced by sexual reproduction, so sperm and an egg joining or a male and female gamete joining, is going to have a unique genome. And remember, the genome is just all the genetic material of an organism. One exception, identical twins. What we're going to focus on then is the process of meiosis today. Now, we need to be able to spell this one because if you stick a T anywhere in there, it's going to be marked wrong. So gametes are produced by meiosis. And I've given you a hint there, E for egg, O for ovum, little reminder about what it's producing. What we make as the end result of meiosis are 4 haploid cells. So that means we're going to get 4 cells with 1 set of chromosomes each. Now, in the case of humans, this will take place in the ovaries and the testes because that's where we make the sperm and the egg cells, which are the gametes, the sex cells. How does this happen then? Well, the first thing is going to be rather familiar to us because what's going to happen first of all, the chromosomes are copied in that process of DNA replication. So DNA unzips, our DNA bases, the free ones that are present within the nucleus, are going to form the complementary base pairs, A's and T's, C's and G's, until we've made an identical copy of every chromosome present. Those chromosomes then line up along the equator in their pairs, and they're pulled to the opposite poles of the cell. The cell then divides in the process of cytokinesis. Now, hopefully we're looking at this and thinking, well, that's literally just mitosis. Yes, it is. So this first stage is identical to mitosis. So a 2-for-1 learning experience. If you know mitosis from paper 1, you know the first part of meiosis in this paper. But that isn't where it ends in meiosis, because at that point we've got diploid cells. We need haploid. So what happens is a second division. In this case, the chromosomes line up along the equator. They're pulled in half. So a single copy of each chromosome moves to the opposite poles, and then the cell divides. So we end up with 4 of those haploid cells. Now, this is a reduction division. They can ask you some little maths-based questions, really. So if we think about it, we start off with our single cell. Okay, we've got our 46 chromosomes present, if you like. We are then going to replicate that. So we're going to copy all of the DNA, and then as a result of that, we're going to end up with 92, because 46 times 2 is 92. We're then going to undergo that first stage of division, which takes us down to 46 in 2 cells, and then it's going to divide again to give us 23 chromosomes in 4 cells. So this is really what happens with the genetic material here, the DNA. So there's going to be an increase, and then a decrease and a decrease. So just remember that in this stage 2, we have the chromosomes lining up along the equator, pull to the opposite pole, cell divides, but there's no replication of DNA in this stage, only before stage 1. In terms of why we get variation when we undergo sexual reproduction and meiosis, then first of all we get this idea of random assortment of the chromosomes. And by that, what we mean is when they're lining up along the equator, we don't know that they're all going to line up, the ones from the father all on one side, ones from the mother on the other. It's completely random how that happens. And each time it happens, it could be a completely unique combination. Secondly, we have a process called crossing over, which we can see in the diagram at the bottom. What happens is those 2 homologous chromosomes line up next to each other, and then parts of them literally cross over with one another. At that point, they actually swap bits of chromosome. So you can see in the third picture there, a little bit of the red chromosome is on the blue one, and a little bit of blue is on the red. They've literally swapped over some of the chromosome with each other. As a result of that, we have a new combination of alleles. And then once we've made all of these individual eggs and sperm cells, then there is the process of fertilisation, which is completely random as to which egg is going to join with which sperm. Obviously, if there are bits that you want to go through in a bit more detail, you can use the video on the main channel there. You can use your revision guide, your class notes, and as always, I do recommend having a look at least a couple of the past exam papers for paper 2, just to get a feel for the questions that can come up. And of course, don't forget to join us again tomorrow for our next Biology Daily Booster.